Hi everybody, welcome back. Today we're going to be discussing DNA replication. So let's first talk about when DNA replication happens. This happens during interphase, and remember, interphase is that part of the cell cycle that's the longest phase, and it has our three subphases, G1, S, and G2. In DNA replication, it's going to happen in the S phase of interphase. The S stands for synthesis. The cell is doing its normal job, but when it decides that it needs to divide, either to make more cells so the organism can grow, or because other cells like it need to be replaced, the cell will enter S phase, where we will replicate the DNA. This is in preparation for mitosis, where the genetic material is going to be split between two daughter cells, and cytokinesis, when the cell splits apart to actually make the two daughter cells. Humans have 23 different chromosomes, or pieces of DNA. In general, all of our general body cells have 2 copies of each chromosome. We call these general body cells our somatic cells. They're different from gametes, which would be our sex cells, sperm and eggs. So anything that's not a sperm or an egg is going to be a somatic cell in the body, and in general, they all contain 2 copies of each of our chromosomes. So if we have 23 different chromosomes and 2 copies of each, that equals 46 chromosomes. For your chromosomes, 1 copy came from the maternal parent and 1 copy came from the paternal parent. So let's say here the green one came from the maternal parent and the purple one came from the paternal parent. These are called homologous chromosomes because they're not identical, even though they contain information to make similar things. For example, these chromosomes might contain a gene for hair texture. The green one from your mother might contain the gene for curly hair. And the purple one, which came from your father, might contain the gene for straight hair. Even though the genes are not exactly the same, they're both coding for hair texture. To replicate the DNA of a somatic cell, we have to copy both of these homologous chromosomes. We make these copies during S phase of interphase. Although we copy them in S phase, they remain stuck together in this sort of X shape at the centromere. So the little knob in the middle is the centromere. And they remain stuck together at the centromere until the cell goes through anaphase of mitosis. Since we created them in S phase, they're going to stay in this format all the way through G2 and all the way through prophase and metaphase. DNA replication happens in a semi-conservative manner. Semi means partial or half, and conservative or conserved means unchanging. So instead of just making a straight up copy, like a Xerox copy of the DNA, we're going to retain half of the old DNA and use it to make a new strand of DNA for every chromosome. You can see here that the old strand is purple. And after it's split away from the other old or parental strand, new base pairs bind via hydrogen bonds to the DNA molecule. These new nucleotides need to be linked together to form a permanent new DNA ladder. When you look at the 2 new molecules of DNA, you can see that each molecule is half old and half new DNA. That's what we mean by semi-conservative. The enzyme that helps us do this is called DNA polymerase. The enzyme that helps us do this is DNA polymerase. Look at the name of the enzyme. First of all, it ends in ase, which tells us it's an enzyme. And we already know that an enzyme catalyzes reactions. A polymer is a string of monomers linked together by covalent bonds. So DNA polymerase is an enzyme that links nucleotides together via dehydration synthesis. In short, it makes new DNA strands. DNA polymerase is different than RNA polymerase in that it can also fix mistakes in the complementary base pairing. It has a proofreading function. So DNA synthesis starts during S phase. the parental molecule is separated into two templates. Unlike transcription, once the strands are separated, they'll never again go back to the other parental strand. We're going to make new strands to fill in the gaps. The new nucleotides bind. They're shown here in light blue. DNA polymerase is going to link those new nucleotides together to form a polymer. DNA polymerase makes covalent bonds and checks to make sure errors were not made. After replication, the DNA is doubled into this X form. The two copies are held together at the centromere, and each of the arms of the chromosome are now called sister chromatids. They are identical to one another because each arm has an old parental copy which was used as a template to make the new copy. Recall that homologous chromosomes are two non-identical copies of the same chromosome in a somatic cell. They were each inherited from a different parent. Sister chromatids are genetically identical and will separate during anaphase in mitosis. Remember that mitosis is the separation of DNA that we duplicated during S phase. There are 4 phases: prophase, metaphase, anaphase, and telophase. This material is covered in a different video, so I'm not going to discuss it here. That's it for today. See you in class.